And okay, recording has started. So my name is Helene Geiger, and I'm chairman of the Adult Ed Committee for Beth El Ho. Welcome everybody this evening. Our topic tonight is A Night of Miracles, COVID-19 vaccine development, and our speaker is Marc Ostro. People keep telling us that we're supposed to follow the science when we're talking about or looking at vaccines, but for most of us, that's actually pretty challenging. Luckily for us, we do have somebody in our congregation who finds this easy to understand and easy to translate for the rest of us. So we were very happy to be able to call on Marc, and we're very grateful that he generously agreed to do this tonight. Let me tell you a little bit about Marc, just to give you an idea. He co-founded one of the first biotech companies in history, led multiple successful drug research teams. He holds 12 US and European patents. He started a venture capital firm specializing in biotech companies. His skill set provides him with the tools necessary to evaluate and advise investors about clinical trial designs and regulatory strategies relating to drug development. So this, he's really the perfect person to be able to lead us through this. I'm going to turn this over to Marc. I'm going to ask you all to please mute, except for Marc, of course, during his presentation. And then at the end of that presentation, Marc will take questions.
Thank you, Helene, for your kind introduction. I'm going to have to put my glasses on because I can't see without them. I have to read my notes. In the old days when I had a really good memory, I didn't need notes, but I'm getting too old for that. Before I get started, I'd like to just go through a few housekeeping items. First of all, just to bring you up to speed, the first vaccine for coronavirus, as probably you all know, was by Pfizer, and that vaccine was approved and injected into the first patient on Monday. Interestingly, my son, who's an emergency room doctor, got vaccinated today and he feels fine. The first dose of Mrna vaccine, which is the second company that's produced this mRNA type vaccine, will probably be given at the beginning of next week. As of this morning, Mrna's vaccine was recommended internally at the FDA for approval and won't be formally approved until the FDA Advisory Panel Committee meets on Thursday. Everybody expects that they're going to give it a rapid approval, and therefore I think the first patient will get dosed Monday or Tuesday of next week. So with having said that, let me say that I'm going to break my presentation into 6 parts. The first is really a bit of housekeeping and background. Then I'll specifically talk about what exactly mRNA is. I know that's probably ancient history for a lot of you who haven't taken a biology course for about 40 years. I'd then like to talk about how the immune response works against a virus. Then talk briefly about the mRNA vaccine and how it's used as a vaccine, and the vaccine formulation, what it's all made out of, and then finally the data from Pfizer's phase 3 trials. The data from Moderna's phase 3 trials is not really publicly available yet, so I won't be able to talk much about that. Although we do know the end result was that both of these trials resulted in a 95% efficacy rate. So having said all of that, let me get started. So first of all, you have to understand what's very special about these 2 vaccines. There are lots of vaccines on the market, but these are the very first vaccines that have ever been based upon messenger RNA or mRNA technology. For those of you who haven't had a molecular biology course or a biology course, as I said, in a long time, I'm going to go into that. But as I said, it's never been used before for a vaccine. In fact, mRNA has never been used before for any kind of a drug. So I guess the reason why Helene asked me to do this is because a long time ago, actually 42 years ago, when I was an assistant professor at the University of Illinois Medical School, I worked on the possibility of using little bubbles made out of fat, they're called liposomes, to deliver messenger RNA into cells. So what I did was I isolated message from a specific cell type in rabbits, and this message was specific for making hemoglobin, or essentially the globin part of hemoglobin. And I was able to show that you could use these vesicles to insert the messenger RNA into cells, and that those cells would make the corresponding protein. Now, I'll get into all of that in not what I did, but how all that works in a second. What I did was extremely primitive. I didn't do it in a human. I did it in a glass bottle with cells. What I did was extremely inefficient. Very, very small percentage of what I tried to get into the cells actually got in. Now, that is absolutely not the case for what Pfizer and Moderna are doing. And in fact, what they're doing is so extraordinary. It wouldn't surprise me if at some time in the future it warrants a Nobel Prize in medicine. But anyway, that's just my opinion. So the things I want to point out sort of as caveats is that I am not an insider. I don't work for Pfizer. I don't work for Moderna. A lot of the details of what they're doing is proprietary. So, and I don't have any access to them. So I suggest that if you really want detailed information about whether or not you should actually go get the vaccine whenever your number is called, I suggest that you go talk to your family doctor. I am not recommending that you take the vaccine. I'm not recommending that you don't take the vaccine. It's not my place to do that. So, last caveat is there's a very mixed audience out there. Some of you are scientists. I can see Sheldon over there in the corner, who is a scientist, and this is probably going to bore him silly. So if you want to get off the call, that's fine. And probably some others of you are going to be a little bit bored. However, the vast majority of you, in speaking to at least some of you, you could probably use a bit of a refresher in how all this works. So I am going to risk boring those brilliant people out there who know all this, and hopefully bring everybody else up to speed. For boring some of you, I apologize in advance. Anyway, so the first thing I want to talk about, as I indicated, is what the heck is messenger RNA anyway? As I'm sure all of you know, all cells in the body and in all living things have DNA in their nucleus, with the exception of red blood cells. They don't have any nucleus at all, and they don't have any DNA. But other than that, all cells have DNA in their nucleus, and the DNA carries the information that differentiates you from a sea slug. I mean, what's interesting is the molecule, the DNA molecule that you have in your nucleus is composed of exactly the same thing that the DNA that's inside the nucleus of the cells of a sea slug or of a tree. Same thing, they're made out of the same chemicals. So the DNA has 4 chemicals, basically, few exceptions, but those are adenine, guanine, thymine, and cytosine. I won't quiz you on that. They're abbreviated A, T, G, and C, but all DNA is made of those 4 molecules. Now, what makes the difference is that how those molecules are strung together. Now, there are enzymes in the nucleus that use DNA as a template, and that template is then able to create another molecule called messenger RNA. So you have DNA in the nucleus, you have a bunch of enzymes that are capable of converting DNA into messenger RNA. So what does that mean? Well, messenger RNA is not a copy of all of the DNA that's in the nucleus. There are thousands of messenger RNAs Each one corresponds to a specific gene, and each gene corresponds to a specific protein. So you start out with the DNA, you have the gene inside the DNA, the messenger RNA is produced as a copy from that DNA, and the message then can leave the nucleus, go into the cell, where it can produce the protein that's specific for the DNA gene that it started from. So hopefully you followed all that, and I'll move forward. Now, once produced, I think I already did that. Okay, so how can you think about this in a more simple way? And I used to have students a long time ago, and the way I tried to describe it to them was that imagine that you had a tape recorder, and the tape recorder had a, for those of us old enough to know what a real tape recorder looked like with the tape and everything, the little brown ribbon, well, the ribbon, you could think of the ribbon as messenger RNA. And then you could think of the tape head, which got music out of that piece of plastic, as the machinery inside the.
We just lost sound.
Sorry, we just lost sound. You just became
Got
muted,
it.
Mark.
Okay, can you hear me? There we go.
Yes.
Touched the button by accident. So where was I? Oh,
You were at the tape head.
I was at the tape head. So the tape head represents the machinery inside the cell, which then can read the tape, which is the messenger RNA, and produce music. Okay, but in this case, the messenger RNA doesn't produce music. It reads through the tape head, which is a whole other mechanism that I'm not going to get into in this discussion. And instead of producing music, it produces proteins. Okay, so you start out with DNA in the nucleus. It produces messenger RNA in the nucleus, which leaves the nucleus, enters the cytoplasm, and there it can produce proteins specific for the gene from whence the message originally came. So hopefully that's a little bit clear. Now, proteins are often called the building blocks of life. I'm sure you've heard that, or maybe not. And substances comprising many of the key parts of the body are made out of proteins. Lots of things are made out of proteins. Enzymes are all made out of proteins. Many of the molecules on the surface of the cell are made out of proteins, and these are called receptors. So lots of things that proteins do, but I'm not going to go into every one of them. Obviously, I'm just going to talk about two kinds of proteins. One is an antibody. Antibodies are proteins, and the other would be a receptor, and most receptors are proteins as well. And receptors are molecules on the surface of a cell that stick out. So think about a pin cushion with pins sticking out, and the pin cushion is the cell and the pins are the receptors. Okay, so I think that's a visual way that you can represent that. So what about the immune response? So how does all this trigger your ability to fight off an infection? Like a vaccine would allow you to fight off an infection. Well, the immune system consists of a whole variety of different cell types. So some of these cell types make antibodies, and they're called B cells. And some are capable of directly attacking a foreign invader, such as a virus, and these are called T cells. Now, these cells the T cells have a lot of different functions. One of their functions is to help B cells in defense against the virus. So those are the only cell types that I want to talk about. Importantly, if you're a healthy person, you have an immune system which can tell the difference between what's called self and non-self. So self is you. So you have antibodies, and antibodies will not be produced against your cells. So that's you. So that's self. The only exception to this is if you have an autoimmune disease. If you have an autoimmune disease, you can produce antibodies against your own cells, or you can produce killer cells against your own body parts. But let's forget that for the moment, because that's not relevant to this discussion. So the recognition that your immune system has for the virus really then depends on the immune system realizing that the virus isn't you. The virus is a foreign invader. So as a foreign invader, the immune system gets revved up. It recognizes the proteins on the surface of the virus as being non-self. It, once again, is not you. And the immune system, thus stimulated, can kill off the virus. So that's how your immune system normally works when you get infected. So how do the mRNA vaccines play into all of this? And as I said, both the Pfizer and the Moderna products are messenger RNA-based vaccines. Now, up until now, a typical vaccine, let's say the flu vaccine, is used by making weakened virus or diluted virus. And often, for instance, the flu vaccine, you grow these viruses in egg yolks. Thousands and thousands and thousands of eggs are infected with the virus grows, and you isolate, purify, and you end up with a vaccine that you can inject for flu, for instance. But these viruses aren't their full infectious self. They're weakened, they're called attenuated, or they're very diluted, so you don't get sick from the virus. So that's a typical virus, been around for decades. That's the way it's done. Now, more recently, there's other ways to make a vaccine. You can isolate proteins from a virus and mix it with other molecules that stimulate your immune system to make antibodies against that protein. I'm not going to talk about those. There are more complicated methods using what's called viral vectors. I'm also not going to talk about those, but I am going to talk about, as I said, the mRNA vaccines. There are lots of pros and cons to all of the above virus vaccine methodologies that I just talked about, but all of them involve complex and or time-consuming manufacturing and purification processes. None of these are present when you make an mRNA vaccine. So the mRNA vaccines have a huge advantage over every other vaccine technology that's ever been developed, in that they can be produced simply and cost effectively. And I'll tell you a little bit more about that. So now that you have, let's say you have the vaccine, how does it actually work? In the case of, let's assume you have a normal coronavirus infection. So you got somebody who's sick. After entering the body, the virus binds. So you have a patient, the virus enters the body. On the surface of the virus is a protein called a spike protein. The spike protein will bind to the surface of target cells. Now, these target cells may be in the lung or they may be in other organs, but when the spike protein attached to the virus binds to the cell, it triggers the introduction, the insertion of the entire virus inside the cell. Now, what happens then? Well, once inside the cell, the virus reproduces like crazy, and it keeps reproducing and making thousands and thousands of viral particles until it bursts the cell. So now all these viral particles that are infectious come out, and they can enter thousands and thousands of other cells. And in this way, the infection gets multiplied and people get sick. So that's normally the way that the coronavirus and a lot of other viruses, for that matter, work. Now, both the Pfizer and the Mrna vaccines use proprietary methods to deliver the messenger RNA that they're using as the vaccine, and that messenger RNA is specific for the spike protein I talked about. So you have a normal virus particle. On the surface is a spike protein, and the spike protein is responsible for the virus binding to the cell. Now, what they've done is they've isolated the messenger RNA that's responsible for producing this spike protein, okay? And they introduce that messenger RNA into the cell so that a cell can now make spike protein, which can be expressed on the surface of the cell where it belongs. The problem is it's damn difficult to get into the cell. Using a typical mRNA, if you added it to a cell in the body, let's say you just injected the mRNA into the muscle, it would be gone in nanoseconds. It wouldn't be around very long. So the key here was to figure out how in the world can we get the mRNA into the cell. Well, I looked and looked and looked because this is something that I was always very interested in because of my work with these delivery systems called liposomes, and I can't find it anywhere. It doesn't exist. And the reason it doesn't exist publicly is because this is the proprietary technology that's being protected by Pfizer and Moderna. So they don't tell you how they do it. We just have to accept the fact that these messenger RNA molecules get into the cell. They have to be inserted into the cell in such a way that they're intact. They can't break down in the process. Otherwise, they won't be able to be used to produce the spike protein. Okay, so let's say, well, not let's say, it does. They're able to get the messenger RNA for the spike protein into the cell. Once in the cell, now there's no virus there, remember? This is an isolated message. There's no other part of the virus, so it doesn't cause any infection. It's totally safe. So they get the message into the cell. The cell makes spike protein. What does it do with spike protein? It puts it on the surface of the cell. That's where it belongs. So now you have a normal cell with spike protein on the surface. So what happens then? Well, the spike protein doesn't belong there. So the immune system recognizes that and mounts an immune response against the spike protein. So now you have the immune system revved up to recognize spike protein from the coronavirus if it shows up. And when does it show up? If you get infected with the real virus. So the real virus comes in, you can't get sick because your immune system is all geared to fight it off before it even shows up. So these types of mRNA vaccines are marvelous. They don't have hardly any toxicity that's been demonstrated so far. Now, you never really know. They've done thousands and thousands and thousands of patients, but they haven't done millions of patients. And until you do millions of patients, you really never know whether there's going to be some low frequency event that occurs. For instance, just recently, after they started giving the vaccine in England, they found a toxicity, but only in patients who were prone to have anaphylactic reactions. Very small number of patients. So these are people with hyperallergies, for instance. So they can rule out those patients when giving the vaccine. But other than that, they haven't found any subpopulation of patients that represent a problem. So that's how the vaccine actually works. Now, what about the formulation of the vaccine? How do you actually make it? And as I indicated, I don't have a whole lot of information on that, but I do have a little bit of information. And I have nothing on Moderna. Moderna hasn't published anything. And even though Moderna is getting reviewed by the FDA advisory panel on Thursday, I still haven't been able to find their document that they used to get approval, but I did find Pfizer's. And in there, I found out that Pfizer is in fact using a lipid vehicle made out of 4 different types of lipids, but they don't tell you what those lipids are. And they don't tell you what the ratio of one lipid is to the other. They don't tell you really very much of anything. So I can't tell you much more about the formulation than that. And as I said, there's absolutely nothing about the Mrna virus in terms of what its formulation is. I do want to point out one thing, that the Mrna vaccine, the Pfizer vaccine, in addition to being encapsulated in some kind of a lipid vehicle, the RNA, the messenger RNA, is slightly chemically modified to enhance its stability and also make it more able to be translated into protein in the cell. So it enhances its stability and allows it to be effective more readily inside the cell. And the other thing is that both the messenger RNA vaccines from Pfizer and Moderna are made in a factory. These are chemical syntheses. This isn't a biological process. They have the sequence of the 4 bases I talked about, the ATGC. They know that sequence of the message, and they can synthesize it in a test tube. So they have a factory that makes this just like it would make sodium chloride. It's just, it's a lot more complicated than that. It's a synthetic process and can be done very rapidly and is automated. So that's really good news for getting out large numbers of doses to the general population. The last thing I want to talk about are the phase 3 clinical trial results from Pfizer. Don't have anything from Moderna. So let's look at the safety first. These vaccines are extremely safe, at least as far as we've looked, which is 90 days after they were first injected. And they're going to keep following these. If they have to follow them for years, they will do that. But the patients used, there were 38,000 patients in the Pfizer study, divided in half. 19,000 were given the messenger RNA vaccine and 19,000 were given placebo. At last look at 90 days, there's absolutely no difference between the two in terms of safety. One is not more safe or less safe than the other. One surprising piece of information, and a lot of us would be happy about this, is that if you're over 56, the vaccines are better tolerated. So I don't know how many of us are under 56, probably not many, but if you're over 56, we're in good shape. The other thing that's really interesting about the Pfizer vaccine is that you get better neutralizing antibody. Well, let me back up a second. When you get sick, if you get coronavirus and you're sick, your body is making antibodies against the virus to try to get rid of it. Now, when you're rehabbing from the infection and you're all better clinically, and you look at the amount of neutralizing antibody, that's antibody that can kill the virus, in patients that have been sick, that level is lower, surprisingly lower, significantly lower than patients that have been vaccinated with an mRNA vaccine. So that's another piece of very good news. On the efficacy side, as has been publicized all over the place, both of the Moderna and the Pfizer vaccines are 95% effective, which is truly extraordinary. I've never seen results that good. Now, these results, if you look at the 30 or the 19,000 patients who got the coronavirus vaccine, it was the same. The results were the same. It didn't matter what your age was, your sex, race, ethnicity, or country of origin. It was always the same. So that's also very good. And finally, the last piece of information on the trial is that after the first dose, remember there are 2 doses, on day 1 and day 21. So on day 1, after the first dose, 1 week after the first dose, there was 52% protection of those patients after dose 1. Obviously, after dose 2, that went up to 95%. But that's also pretty extraordinary. And the last thing I want to say before I open it up for questions is that there is a difference in convenience between the Pfizer and the Moderna vaccines. The Pfizer vaccine has to be stored at minus 80 degrees centigrade, which is minus 112 Fahrenheit. That's dry ice temperature. Whereas the Moderna vaccine can be stored at minus 20 degrees centigrade, which is no different than just putting it in the freezer in your kitchen. So having said all of that, I'll shut up and open it up for any questions that you might have.
I guess if you do have questions, either raise your hand electronically with the little raise hand thing, or just raise your hand in front of the camera and we'll try to pick this up and call on you.
Did I lose everybody?
Lauren and Flo have a hand up.
Yeah, hi, Mark. Thank you for this. The question I have is regarding the temperatures that the vaccines have to be stored in. Can you explain why there's
Why there's a difference?
Right.
If I knew the formulations of both, I could explain it to you. It's all formulation based. The messenger RNA should be stable for a while at -20 all by itself in ethanol. But given that I don't know what the formulation is, I can't tell you anything about that. I wish I could, but I can't.
Thank you.
Additional questions?
Dad, I had a question, if I could chime in.
Sure.
So I guess my question for the vaccine is how, I guess I don't know the half-life of mRNA and how long after you are given the vaccine does your cells actually produce this spike protein? And at some point, is there, like, the spike protein will no longer be produced? So I'm just curious how long your immune system will be kind of, because presumably it will be attacking your own cells if it's, you know, attached to the surface of your cells. So like, how long does that process take, or do we not know, or is that proprietary as well?
It's not proprietary. How long do you want to know how long the spike protein exists on the surface of the cell, or how long the immune response against the spike protein lasts, which is the most important?
Yeah, I guess, you know, how long will your body, the cells be producing the spike protein? I guess is kind of the bigger question that I have. I guess I don't know if we know.
I don't know how long the cells are producing the spike protein. That wasn't in any of the information that I read. What's the most important in terms of duration isn't how long the cells are producing the spike protein, but how long, once the immune system is sensitized to the spike protein, how long it remains sensitized and will be able to recognize an invading virus. So that's the longevity that matters, not how long the spike protein is present on the surface of the cell. Does that make any sense to you?
No, that makes sense. I guess I was thinking more from a toxicity standpoint in terms of if your body continues. That's what my question was, but I'm not sure we know the answer to that.
Well, you know, we don't know the answer, but we do know that so far, after 90 days, there's very little toxicity. We don't know how long that's going to last. That's the problem. You don't know how long until you know how long. There's no way to shorten that.
Helene?
Yes, I can't see who's speaking.
Kara, I have
Kara.
a question. You said about people who are immunosuppressed. So if you have somebody that has an immunosuppressive disease or a lupus, is there an increased risk for taking the vaccine?
That's a damn good question.
Thanks.
I don't know the answer to that question. I'm not sure anybody could give you an absolute answer to that question. And I think it would depend on how you're immunosuppressed, which cells are involved. So that's a very specific question that I can't answer. And I would only recommend that you ask your immunologist or your rheumatologist, whoever you're seeing, that question. And I'm not sure they're going to be able to answer it definitively either. This is, you know, typically when a vaccine is approved, there's a heck of a lot more data available because they're taking years and years to develop it. This was developed very quickly. So a lot of these questions that you're asking ultimately will be answered, but it's a little difficult to answer based on the data that's currently available.
We have a question from the Boonans. Will we need to take this vaccine annually, or will we just need to take this once?
Well, as I said, we don't know how long the immunity lasts. So it's very possible you're going to have to take it every year, just like the flu vaccine. It's also possible that you get long-lasting immunity for longer than a year, but you don't know if it lasts a year until you wait a year. I don't know how to accelerate that.
Okay. We also have a question from Warren Bilker. Is there information about the potential array of strains of COVID-19 and if the vaccines protect against all of the strains known at this time?
Well, so far it does protect against all the strains. That's not to say that there won't be a mutation at some point that allows that virus particle to escape the vaccine. But as of right now, given the broad number of patients, if you add up the number of patients between Pfizer and Moderna, it's almost 80,000 patients. And so far, it's been effective in 95% of the cases. And you would expect, I don't have the breakdown of all the different strains in these 80,000 people, but certainly there's more than one strain. And it's been effective at 95%. So we'll just have to wait and see whether new strains arise that generate resistance to the vaccine. But at this point, so far, so good. Let's put it that way.
Other questions?
Sheldon.
Sheldon?
Yeah, Mark, do you know if any of the other vaccines are weakened virus or attenuated virus or killed virus?
Of the ones that have been publicized, none of them are using standard attenuated or killed virus vaccines. They're either the mRNA vaccines or protein-based vaccines or adenovirus-based vaccines. I'm not aware of any that are using just typical egg incubation techniques. The J&J vaccine, for instance, is an adenovirus-based vaccine.
Well, I have just a really stupid question, which is, would you take this vaccine?
Oh, if it was there tomorrow, I would stick my arm out. No question about it. My son, as I said, my son took it this morning, but he's an emergency room doctor, so he didn't have any choice, but he would have taken it even if he did.
No, I had a choice. It was voluntary, but I chose to take it.
Oh, you're on. That's right. You're on.
We have a question from Alex.
Mark, how did they determine the dosage? And, related, why are we getting 2 shots as opposed to 1? And, how does the dosage determine that we have just the right amount of immunity, not an overreaction, immune overreaction or underreaction?
Good question. What I talked about in terms of clinical trials was the phase 3 trial. When you get to phase 3, typically you've done a bunch of patients in phase 1 trials and phase 2 trials. And the purpose of phase 1 and phase 2 is to determine what the dose should be, what the dosing schedule should be. All those kinds of questions are answered prior to starting a phase 3 trial. So those questions are asked, and hopefully they were answered in the phase 1 and 2 trials that Pfizer and Moderna conducted.
Thank you.
Thank you. We also have a question from Sharon S.
If you have had the disease and you've recovered, will those people be taking the vaccine, and is it safe for them to do so?
Yes, they will take the vaccine, and my son just piped in, but he got coronavirus and he took the vaccine this morning. And he's, as far as I know, he's still up and running.
Okay, any other questions? If you have, please, oh, Charlotte, you're muted.
There we go. Did the studies include or exclude people who are immunodeficient? Do you happen to know?
I think they were excluded, yes. Which makes perfect sense. If you were developing a vaccine, you wouldn't want to use it in somebody who had a compromised immune system, because that would be a big caveat.
Should you take the medicine or should get it?
Mary has a question.
Hi. So thank you so much for this. I was hoping you would elaborate on the fact that, so the vaccine doesn't prevent a person from being exposed and getting coronavirus. It allows your immune system basically to fight it. So could you address the whole issue of that person who's vaccinated happens to kind of get corona but fights it, but their status as contagious?
Well, once the virus, they're not going to be contagious. Once the virus is killed, it's not going to infect anybody. And that's what happens if you have a functioning immune system and you're mounting a strong response against the virus. So dead viruses don't infect anybody.
But wouldn't there be a period where maybe that person might test positive for the virus, even though their immune system is fighting it and they're not coming under ill effects? But I'm just wondering how this is all going to work with
Well, in
masking
fact,
and.
If you looked at in detail at the write-up that was submitted to the FDA from Pfizer, in the first week or two after the initial injection, some of those patients actually got infected. They actually got infected, but the severity of the disease was minimal. They didn't really have a significant problem. But there were some patients who actually got infected while they were in the trial, but only during the first period between the first dose and the second dose, minus the 5%, because it was 95% effective. So 5% did get infected and got sick.
Other questions? I don't see anyone else. If you do, please unmute and just
Lane,
pipe up.
there's another question in the chat.
Oh, yes. The lower half dose was more effective for the Pfizer vaccine. How can this happen?
It wasn't the, first of all, it wasn't the Pfizer vaccine. It was the AstraZeneca vaccine. It's a genuine conundrum how that can happen. They've got a big problem because, first of all, there were they did a total of 20, I don't know, about 21,000 total patients, of which 3,000 got the half vaccine first and the full dose second. And that's where they had the 90+% response rate. In the other group, they got a 60% response rate where they got full dose. And you have to think about that for a second, because the patients in the 3,000 dose group were all under the age of 55. And we all know that patients under the age of 55 don't get very sick and don't get infected as frequently. So I don't know what that means. What it does mean, and it's bad news for AstraZeneca, is that they have to redo the study. And, you know, it wasn't intentional, for instance. The half dose, full dose was an accident. They thought they were giving full dose, and it turns out that in those 3,000 patients, they gave the first dose and then, oops, we blew it. We gave a half a dose. So they decided to continue and give the full dose after that. So there's all kinds of questions that come out of that. And as a result, that data is useless. They're going to have to redo it.
Other questions? Lauren, I think your thing is still your hand is still raised, but that's from your previous question, right?
Yeah, that's right.
Yeah,
Previous question. Sorry.
Who has his hand raised?
Warren, did you have another question?
No, I didn't. I'm not sure why my hand is still raised.
Oh, okay. Sorry.
Getting tired.
Okay, well, I think that's everything. And I want to thank you so much, Marc. This was wonderful. This was amazing. All I can say is thank you very much for a wonderful presentation and so much information. And we were really blessed to have you.
Well,
So thank you.
pleasure. I'm glad to be able to do it. I hope it was useful for you. Not a waste of an hour, at least.
Not at all. Oh, I'm sorry. Sharon has another question, I see.
Just wanted to know if pregnant women will be taking the vaccine.
I don't think so in the very near future. I think they're going to need more data before they're going to give it to pregnant women.
Thank you.
Alina, I think.
Charlotte, I see your hand is
has
raised.
also a question.
Okay, Charlotte, and then
Just one quick question. I'm kind of curious. Is there anybody willing to say whether they would, on this call, willing to say whether they would or wouldn't get the vaccine? I was wondering what the consensus was.
I would take it.
I'd take it in a heartbeat.
Okay. No, I can't.
Sign up.
Izzy, I see you have your hand up for a while.
Yes, I have a technical question. Some of the vaccines are more effective, more potent when they're given intradermally, like rabies. And this vaccine is inexpensive. So could you increase the dose or give it intradermally and get by with one injection?
It's a good question. I was unable to find anywhere in the literature an explanation as to why it's given intramuscularly as opposed to subcutaneously or intradermally. I didn't find anything. The only conclusion I can come to is they did a lot of work in animals and probably found that the best immune response was IM, but that's just a guess. There's no literature that I was able to find that would explain that.
Yeah, I was interested. Is he from your synagogue or?
Yeah.
Thank you.
And I think, Mary, you had your hand up?
I just wanted to clarify that Izzy is here. He's my friend, and I invited him. I invited my friend Flossie, and I invited my friend Sharon S., who asked the question. Oh, and there's Izzy's wife, Ella. So welcome to our community. We're so glad to have you, and we're so glad that this topic was of interest to you.
Absolutely. Absolutely. You're all very welcome. Any other last questions? Ruth Lax, who's muted.
Yep. Okay. Yeah. Thank you. Yeah, Marc, I was just wondering, with your range of experience with this, given that the drug companies and the FDA was really pressured by the administration to get something out, Project Warp Speed and all that. There's no reason for us to be concerned about the safety of the drugs coming out.
Not in the least. I would not be concerned. I've been to the Food and Drug Administration at advisory panel meetings 3 times. And trust me, these people are, what happens when you get 20 people who are all experts in a room at the same time? They all want to prove how smart they are. and they all want to be as negative as possible so they can show how smart they are. So believe me, if you get a 17-4 vote, which is what Pfizer got, that's extraordinary. I don't think I've ever seen a 17-4 vote. Having developed a drug which got nixed at the FDA years and years ago, I can tell you it's not easy, and getting that level of support is amazing. These people do not succumb to political pressure. They don't give a damn.
Okay.
And the FDA, except for the head of the FDA, they're all lifetime, they're lifetime employees.
Okay.
They're not political appointees.
Okay, thank you.
Excellent.
To add a comment to that also, doing this kind of work. The phase 1 was a little longer than usual on these trials, and they were able to get the right dose in phase 1. That's why they were allowed to skip the phase 2. They accomplished both phase 1 and phase 2 in phase 1. I think Marc already said that. The other way they rushed the process was the federal government both funded allowing the assembly lines to get started at the beginning of phase 3 to go into production and storage. This is highly unusual in that usually the companies pay for that development and they don't do it until after phase 3 is approved. And the reason is not a safety reason, it's a business risk of bearing that expense. So by the federal government taking that on and allowing them to start as soon as their phase 3 trial was approved to proceed, that was a real gain in time.
Yeah, absolutely true. Thank you, Eva.
Talk very much, Marc.
Any last minute questions? If not, we will release Marc from.
Okay.
Marc, this was great. Thank you so much.
All right.
I really appreciate it.
Thank you for having me.
I'm going to stop the recording.
Okay. Good night, everyone. Have a good evening.